Thank you. All right. The test is 2 weeks from this Friday. And as I just said, we will only meet one class next week. So there's this class and Friday, one class next week, by which I hope we get done with chapter 18 as well as chapter 17 that we're going to finish today. And then after next week, we'll have a week where it's class, test. So we have another test coming up pretty quickly. Make sure you're keeping up with your reading. What we talk about today is your reading for tonight into next class. We will have an activity either at the end of today or next class. It's already posted on the coursework page. And I do intend to have some AP Classroom problems for you as well to start reviewing for the test and for more grade. More gradage. So last class we talked about transcription and translation, which is how you go from DNA to mRNA to a ribosome where proteins are made one amino acid at a time based on codons that are in the RNA message. We went into a bit of detail about transcription, about RNA polymerase, separating the strands, writing a strand of mRNA based on what the DNA template strand says, and then how like introns are cut out and the exons are glued together, and you finally process it down to the final message that the nucleus is sending to the ribosome. Today, I want to look at the translation part, the reading of the message. And your book goes into a lot more detail than I'm going to hit on today, but I'm going to try to hit on some detail. And there's some really crazy sounding vocabulary that I want to make you aware of. And then we're going to finish with like 6 slides that I'll go through pretty quickly on different types of DNA mutations and what they can result in. So we're going to talk about translation and mutations today, and then that will take us through the big stuff in chapter 17. So right here, you see a pretty comprehensive picture of translation. We've already written out our mRNA molecule, written out our message here. You can see this is the mRNA, and it is in a ribosome, which is this weird kind of glob right here. We've talked about ribosomes plenty of times. We mentioned them way back in first semester as the site of protein synthesis. Today you're going to learn more than you want to know about what that term protein synthesis means. One thing I want to start with, though, is that the way ribosomes are able to make proteins based on this message is with another kind of RNA, and it is called tRNA. The T stands for transfer. So while mRNA is the message, tRNA is the chemical responsible for transferring or simply bringing amino acids, shown here in purple, to the ribosome so that you can stick these amino acids together as a chain of many amino acids known as a polypeptide. So tRNA, which is shown in kind of this light blue, or I call it antique teal, they are the structures bringing the amino acids to the ribosome. But there's a lot of talk about when we say tRNA brings the amino acids to the ribosome. So I want to look at tRNA in more detail, how these tRNA molecules get the right enzyme, and before we get there, how the tRNA even recognizes when it should hop in and drop off the amino acid. Which brings us to a term that you saw, and I told you to scratch out on the last little sheet that you'll have time to wrap up today if you have not finished it. The term anticodon. So mRNA has these three-letter segments called codons. The way a codon works is that a codon attracts an anticodon that matches it. So here we see an error. This is actually not accurate. But this is a good example. This UGG codon attracts a matching or complementary ACC. U's and A's are attracted to each other. G's and C's are attracted to each other. We've talked about these base pairings before, and we see it again here. So there is a three-letter sequence on the tRNA called an anticodon, and it is what matches up with the codon. What's the problem right here? Yeah, it should be AA for the UU, but this G should attract a what?
C.
A C. So that is a typo from your book that I just caught just now. And there's actually another one right here, but be aware of that. So let's take a look at what exactly tRNA is. This is how you typically will see a tRNA molecule shown. It has this weird like clover leaf look to it, but it is just a single long strand of RNA. And what are these dots that are representing intermolecular attractions called?
Hydrogen bonds.
They're hydrogen bonds. There they are right there. So thanks to the same hydrogen bonds that you see that hold the double helix together, these same hydrogen bonds are going to be giving this tRNA this specific shape here. Now, as you saw in the previous slide, you can represent a tRNA as this little like asthma inhaler or whatever shape this is. This is how your book frequently simplifies the drawing. And as you can see, you have A's and C's and G's and U's. So you could look at this and just tell me it's RNA because it has uracil instead of thymine. But at one point on this structure, you have a three-letter sequence. Of all these letters, there's that single three-letter sequence at this point called the anticodon. That is the three letters that will complement the three letters of each codon on the message. So this anticodon is, as we saw in this previous slide, just these three letters that match the three-letter codon in the message. Now, you can look at it in this more complex ribbon appearance, but that's not really a helpful diagram for our purposes. Really, here is all we need, because the tRNA at this point in your understanding of biology really just has two important parts. The anticodon, and then the place where it can hold and carry and then release an amino acid. So those are the two things you want to really focus on with a tRNA or remember about tRNA, is there's an anticodon and then a place for an amino acid. Now, your book continues with a discussion of how is it that a tRNA can even get an amino acid. So a tRNA with a specific anticodon needs to have an amino acid attached to it before it goes to the ribosome and participates in the building of a protein. And this is the enzyme right here in blue, and it's also represented here and here. This is a class or a family of enzymes that have a wonderful name. The name is aminoacyltRNA synthetase. Isn't that a fun, simple name? But in all of this, I mean, this looks like some of the unpronounceable things that they have in like the ingredients list of a bag of Cheetos. But as crazy as all this looks, how do I know from this name that it's an enzyme?
Amino.
ACE.
Oh, sorry.
ACE. Now, how do I know it's an enzyme that deals with amino acids and tRNA? Yeah, we see amino here, we see tRNA here. What is the syntha mean?
Fake.
Synthetic sometimes is a term we use to mean fake, but it really means to build. So you're building or combining a tRNA and an amino acid together.
So
That's the function of this enzyme.
synthetics, like even though the dictionary definition is fake, but like in reality, like the root synth is for building.
Yes, great point. The word synthetic or synthesis, it has a connotation of meaning fake in kind of today's society, but it originally meant to build or create. And here's the thing. The reason it means kind of fake now is that you have naturally occurring food and materials and chemicals, and then you have ones man creates. And those are the synthetic ones. So since people make some of these chemicals in our environment, you could see it as being fake, but it just means we synthesized it.
Yeah.
That's all it really means. I've never thought about that, but that is a kind of a conceptual issue that you could have. Now, like any enzyme, there is going to be an active site, and this enzyme, these aminoacyltRNA synthetases, are going to have an active site that recognizes a specific anticodon and a specific amino acid. So it's this enzyme that matches kind of anticodon to amino acid, and then the right or the correct tRNA is going to be holding or carrying the correct amino acid. Now, like I said, this is a family of enzymes. So when you do your reading, they will talk about these aminoacyltRNA synthetases, but they will also use a specific name, like in this case, you have tyrosyltRNA synthetase. This is the specific enzyme that takes the specific amino acid tyrosine and puts it to the specific tRNA that it needs. And the reason I'm saying the word specific so many times isn't just because it makes my mask feel gross, but it's also because enzymes give us this specificity that we need. How do we get a specific tRNA to hold a specific amino acid? Well, you have a specific enzyme that ensures that reaction between those components. By the way, what does this mean? In general terms, it needs energy. You're using some energy to bond them together. Now it looks a little different because ATP is being converted to AMP. We're turning triphosphate into monophosphate, which means we're actually pulling two phosphate groups off of the ATP to do this. That is not really important, really. I mean, it's a reaction, it needs energy, that's kind of fundamental stuff. I would never, on a test, give you some kind of like enzyme, amino acid, tRNA, and show them all combined and be like, fill in the blank. I would just have this on here and maybe, just be able to recognize in diagrams that this is an indication that energy is involved in this process. So it is an energy consuming process to do this. Of course it is. We're building order. Okay, so once the right tRNA is holding the correct amino acid, then it's ready to travel to the ribosome. And what you're seeing here is essentially the same thing as from the first slide, except there's a little more detail thrown in here. So this is like it says, a computer model that shows a more realistic view. The ribosomes are very lumpy and clumpy looking. You can see a chain of amino acids. This is the baby protein that is forming from the process, and you can see this chain, which is the mRNA, being read by the ribosome. Ribosomes are broken into a small piece and a big piece. They're two halves, and they actually come together around the message. And your book will talk about the small subunit and the large subunit. It's kind of the small half and the big half that come together. Not terribly important, but it's something we will see later. So again, and a lot of what I'm doing is repeating here, the message comes through the ribosome, and one codon at a time, a tRNA will show up, recognize a codon, and then drop off the amino acid it's carrying, and then eventually it will release and a new tRNA will come in. It's a conveyor belt of sorts, where as the message goes through, tRNAs arrive, drop off, and exit. And there's 3 locations that your book refers to within a ribosome, which is the A site, the P site, and the E site. And they have those letters for a reason, but this is how I would remember them. There's a site where a tRNA arrives, and then it puts the amino acid into the chain, and then what does it do? It exits the location and goes off to be used again later if it needs to. So there's kind of an arrival location, a location where it's patching a polypeptide to this petty protein. Just trying to alliterate there, sorry. And then it exits. And then it exits.
That was a good attempt.
Thank you. Thank you. I try. Now, I want to go back real quick, not to kill a bunch of time here, but I think this is kind of neat. The message is being read from a 5' to a 3' direction, okay? So we're 5 to 3, 5 to 3. The anticodon is arranged in what direction? Very good. This is the 3 end, and if you follow it around like you're playing a weird board game, this is the 3 to the 5 end. So it's always this weird anti-parallel thing that I just want you to kind of be aware of. If the message is reading 5 to 3, the anticodons that are reading that message are from 3 to 5. The anti-parallel nature of these nucleic acids always comes up. It just kind of helps simplify some of the notations you see on these diagrams. Okay, so anybody remember the start codon? What was the three-letter sequence? AUG, good. There it is right there. AUG is the start codon for this process. So what ends up happening, again, kind of delving into a little more detail here, translation really begins when the message just kind of lays itself down and attaches to the small piece of the ribosome, the small subunit. And at some point along this message, hopefully, for the cell's sake, there is an AUG, and that becomes the location where the first tRNA bonds, and it's always going to bring methionine. That's kind of your starting amino acid that's coded to AUG. That's the point that triggers the large subunit to lock into place, and then you really have your ribosome. By the way, any guesses as to why we call it a ribosome? Where do we see this ribo come in? Yeah, it is from ribonucleic acid, or RNA. Ribosomes are actually made of RNA as well. So there's messenger RNA attaching to a ribosome made up of RNA, attracting tRNA. So this is what RNA does. It's the working tools and equipment that basically read and carry out the instructions of DNA. So here, and there's really nothing new here, it's just another diagram from your book that's showing this process of translation. You have the starting codon that comes in, you have the next codon arrive at the A site, its amino acid is going to be added to an ever-growing chain of amino acids, and then eventually it works its way over to the exit site and is ejected, and new tRNAs will come in. Again, don't really need to know much about this, but what's happening here? What is this in general? You're using energy to drive this process. So a lot of what I try to do in AP Bio is show you these scary diagrams and just try to point out, the more fundamental concepts that they are showing. So energy is being used, tRNAs are arriving, adding, and exiting. And the process just goes along until what? What in the message will stop it? A stop codon should be there at some point. And that's what this next slide is showing. Eventually, a stop codon is going to attract something called a release factor, which causes the ribosome to break apart, release the message, release the polypeptide, and the process is done. So the stop codon causes kind of everything to disassemble and end the process of translation. And the effect that has is your polypeptide chain is now free and nothing is being added to it. It's done. Okay, so that's translation. But there's maybe two more slides that I want to point out some tangential information about. What were the two types of ribosomes that we talked about a long time ago? They actually correspond to the two types of ER, or endoplasmic reticula. There is smooth and rough ER because there are either ribosomes attached to the ER or not. And in relation to that, ribosomes are either free, meaning floating around in the cytoplasm, or they are attached to an endoplasmic reticulum. And your book talks about what determines that. Now again, this might look like a lot of information and look very scary, but here's what we have going on. We have a ribosome reading a message, and it is making a protein. Now here's the thing, there's a lot of things proteins are used for. Some proteins are used within the cell to drive whatever process the cell needs to have occur. Some proteins are used within a cell as just building blocks for cell structures. But some proteins are secreted from the cell. They are packaged away in little, what were the name of the fat bubbles? Vesicles, good. Some proteins are packaged in vesicles and exported and secreted from the cell. Your book talks about insulin. Insulin is a protein. Your cells make it and then they release it into the bloodstream. And insulin is a hormone that travels throughout the body, helping with the metabolizing of sugars that you eat. So every ribosome kind of starts out as what we would call a free ribosome. The process is going to happen in the cytoplasm. But if the protein needs to go to the ER and then the Golgi body for export, it's going to have some kind of amino acid sequence that's called a signal peptide. That signal peptide will attract a special chemical, which here is abbreviated SRP, signal recognition particle. You don't even need to know what it stands for. But there will be a signaling site on the protein that the ribosome is building, and this SRP molecule will lock onto it and carry it to the endoplasmic reticulum. And as you can see in this diagram, ribosome is just making the polypeptide chain. This particle is going to lock on and carry the whole thing to the outer membrane of the ER. And then that growing protein, that growing polypeptide chain, is going to just kind of grow into the ER. The ribosome just ends up kind of pooping it out into the space here inside the ER. So, as the protein is being built, other chemicals can carry the ribosome as it's building that protein and attach it. And so when you see rough ER, what you are seeing is just a place on the endoplasmic reticulum where ribosomes have been brought and attached in order to finish producing the protein that can be left in the ER, later packaged and secreted, and then the ribosome will just break off and go back out in the cytoplasm. So it's one of those things where, like, early on in the year, we were just talking about, like, free and attached ribosomes. Well, you really just have this constant fluctuation of ribosomes are in the cytoplasm and some that are escorted to the ER if the protein needs to be in the ER and needs to get ready for secretion. So insulin would do this. Your body needs insulin. It sends the message to the ribosome to start making insulin. Part of the insulin protein attracts these particles that carry the entire package to the ER, and then the insulin ends up being formed or completed in the ER and sent out. There is a trick that your book talks about that some cells engage in, which is you can have one message with a lot of different ribosomes all attached to it. What does this allow for? What would having 4, 5, or 20 ribosomes all attached to a message, all reading it, let the cell do? Make it produce more.
As an enzyme.
Which, whether it's an enzyme or a protein that has some other non-enzymatic function, the more ribosomes you can attach to the message, the more protein you can make with that message. So this is a little kind of ancient strategy that all cells take advantage of, where you just bring more ribosomes in, more factories for more production. You just have to make the one message, and you can get many proteins from it. And then the next slide is just kind of the entire picture of everything we talked about over the last 2 classes together. Where you have DNA, the mRNA message being made through transcription. Oh, are those people in the hallway? That was so. Those voices seem so close. I thought I was hearing them.
They're at the fridge.
Remember, the RNA that's made at the beginning, there are pieces that get cut out. The pieces are then kind of glued together, and the final process message leaves. It leaves and attaches to a small subunit. Once a start codon is read, the big subunit locks into place. tRNAs enter, add their polymer or add their peptide, exit, and this protein is made either in the cytoplasm, or if the ribosome is attached to the ER, it's made into the ER and later sent out of the cell.
Quick question.
Yes.
Can you sort of simplify the basic purpose of transcription versus translation?
Yes. So here's the thing. The ribosome is just a machine that can make proteins, but it doesn't have any instructions. So you have to give the ribosome the instructions it needs to make a protein. Well, where are those instructions stored? Where's that information stored? That's what the DNA is for. So basically transcription, and that's a great question, and sometimes I do get a little worried that I just dive so much into the details that we miss the big picture and the basics, yeah. But the DNA is the instructions for making, it's the cookbook. It's the instructions for making the product. The message is how you get those instructions from DNA to the ribosome, which actually does the work. Once the ribosome, and that's transcription, writing out the instructions based on what the DNA says. Translation is simply the message being read and the protein being made. So transcription is writing the message. And when I say message, I mean messenger RNA. You're making the messenger RNA. That's transcription. Translation is you take the messenger RNA and you make the protein.
Great way to sum it up. Thank you.
Yeah, well, it's a good question. Okay, now I know I've been talking for like 35 minutes, but I've got one more topic, which is mutations. What happens if you change the DNA?
Mutation results.
Well, any change in DNA is called a mutation, the results of which can be the message is changed, so the codons are changed, So different amino acids are brought in. And if you're building a protein with different amino acids, it's going to be a different protein. Okay? So I have 6 slides, but we're going to spend like 1 minute or less on each one. So there's 6 slides, but I plan to kind of get through this. And each of these slides is an example of how if you change DNA by 1 letter, you can change the message and the protein that you end up making. Maybe a little bit, or maybe the change doesn't matter at all, or maybe it's an absolutely catastrophic change.
Hey, can I interrupt real quick?
Yes.
I mean, I'm going to, but can I get a hammer for Miss King?
Yeah, I think it's in
In that,
one of
Tool
these
drawer?
tool drawers, yes.
Thanks, man.
You're welcome. Thanks for walking in front of the camera.
no problem.
So, in this example here, and in all these examples, we have what we call our wild type DNA sequence. This is our DNA sequence right here that we're going to go with every time. Notice it starts with TAC, T-A-C, and the template strand is going to be this top one. So we're going to be reading the top strand every time. This TAC will be transcribed into a message that says AUG, so that's going to be our starting message. So here we go from DNA to the RNA to, after translation, what amino acid will go with that codon. And this is what we did in that activity last class, where I gave you some DNA, you wrote out the mRNA, and then you found the start codon, started there, and every 3 letters after that was a different amino acid. So we're going to pretend this is normal. This is your wild type or normal common DNA sequence, and the protein, this isn't the whole protein, but the protein should read like methionine, lysine, phenylalanine, glycine. So this met, lis, phi, gly. There are a lot of ways you can change a message. So there is one type of mutation called a substitution. Substitution means you change a letter. Substitution just means you change a letter. Now, one thing that can happen when you change a letter, a type of substitution mutation, is called a silent mutation. And I'm going to show you what's happening here. You can probably see it. But what we're going to do here is, instead of having a G at a point, sometimes mistakes happen. And when the DNA replicates, or you make a copy of DNA, a G could change to an A. So we're substituting A instead of G. Well, in this case, this A will transcribe into mRNA as a U when it was a C. But if you look up on your chart, which you don't have to do right now, but that chart we were using where we basically translate codons during translation, GGU is the same amino acid that GGC is. So this is a case where you could change a letter in the DNA, it changes a letter in the message or messenger RNA, but it's actually the same amino acid, so you wouldn't even know. And that's called a silent mutation. That's one possible mutation. It's a change, but it doesn't end up changing anything. Okay. What else could happen? Again, a substitution. This C here, we're going to change to a T instead. The T, when we write out the messenger RNA, will give us an A at this codon instead of a G. So instead of GGC for glycine, it gives us AGC, and that is a different amino acid. So here, our change or mutation has caused an amino acid to be different. One amino acid is now different. Now, a protein could have 50, 100, or hundreds of amino acids, so changing one amino acid might or might not be a big deal. But we have resulted in a change here, and we call these mutations missense, which is a weird word. Missense. So if a substitution causes the amino acid to change, we call it a missense. That may not have any effect, but there are some really serious genetic conditions that happen because one protein with 150 amino acids has one amino acid changed. And all of a sudden that protein does not work correctly, and it can be disastrous. So missense, could be bad or not. All right, a third of these mutations. Again, here's a substitution where instead of a T, we now have an A, and that A, instead of coding for an adenine in the message, is now a U. And UAG is a stop codon.
Oh no.
So now, and you saw this on that worksheet from last class, now instead of having possibly hundreds of amino acids being made into a chain that folds into a complex protein that serves some important role, now we just start, we have methionine show up, and then we stop. And basically, you don't make that protein at all. This is serious. This can cause big time problems in your body's physiology and chemistry. And when you have what we call a premature stop, that's called a nonsense mutation. Which is kind of a funny, odd word to use, but it's the word we use. So nonsense mutations are the result of substituting a letter, and instead of just changing one amino acid, you stop and you don't get any of the others. Okay, 3 more mutations. Those 3 were substitution. The letter gets changed. These are the biggies that can really cause big problems. You can have what are called insertions, where you actually add a whole new letter in between 2 other letters in the sequence. So here, we didn't change a letter, it's just between the C and the T, we added an A. And I understand that spells cat, but don't say that. Well, here's what happens when you add a letter. You read the first codon, and then when you add a letter, you have what we call a frame shift. Adding a letter means every codon has been shifted over. So every codon after it can be affected. In this example, shifting all the letters down happens to make a UAA here, which is a premature stop. So it says here a frame shift causing nonsense, meaning we added a letter, it shifted our code by 1. Every codon would be affected after this. the reading frame is affected. And in this case, it ends up just like the last slide where you have a premature stop signal, which we call a nonsense mutation. But a nonsense mutation caused by inserting an extra letter. Just like you can add a letter, you can lose an entire letter. So here, where we had C-A-A-A-C, we now have C-A-A, took out an A, and instead of 3 A's before the next letter, we have only 2. Well, again, every codon after this deletion is going to be shifted by 1, so every codon is affected. And so what happens when you insert or delete is before the mutation, everything's normal, and then after the mutation, you might have every single amino acid be different. I mean, you could be making a drastically different protein. Now, they use the term extensive missense because not only is the phenylalanine amino acid different, glycine has been changed, and if you had 100 more amino acids, every single one of them be different. That's a big deal. So it's really bad if these mutations happen early on and affect everything after it, versus if they happen at the very end of the gene and at the very end of the message and maybe only affect a few codons. So where a mutation happens and what kind of mutation it is can be a big deal. And then finally, you can have more than one letter missing, or even more than one letter added in. This is an example of an entire codon that gets pulled out by accident. Obviously, this would probably be more rare than just one letter. It's kind of interesting they were saying there's no frame shift. If you pull a whole codon out, at least the 3 letters after 3 letters after 3 letters are the same after it. But it would result in a change. Any of these changes are known as mutations. It's probably the longest lecture I've given this year. Not my goal, but it does get us through the big parts of chapter 17. And there will be much less to talk about Friday and the one day we meet next week, and we can spend more time on test review. So we're going to do the mutations activity next class, so don't worry about that. Make sure that transcription and translation, just kind of decoder sheet, is turned in today. So get that to me. You can stop the video.